We have with us tonight Dr. Richard Klein. He's an associate professor in the School of Earth Environmental Science at the University of Texas in Rio Grande Valley. He is a behavioral ecologist who strives to apply his understanding of animal behavior and ecosystems to understand, mitigate, and hopefully solve real-world problems, especially those caused by humans. He's been studying the fish and fisheries of the Gulf Coast of Mexico for over 20 years. And since moving to the Valley in 2011, he has focused on fisheries and wildlife issues along the Texas coast. I also want to indicate that he has also helped me tremendously with monitoring of the wildlife crossings on SH-100. So it's with great pleasure I present Dr. Klein.
Okay, thank you, Robin, for that introduction. So I've been asked to give you a presentation on ichthyology. I'm gonna cover a few other topics as well tonight. I would love to be there jumping around in front of you because that's how I present best, but we have to be constrained to our chairs and and online for right now. I will now share with you the presentation slides. Let's see here. Okay, hopefully. I can see. Here. Always got to get everything set up. We got to swap the presenter view. Get it. There we go. Okay, so can everyone see this? The title page? Okay.
Yes.
Yeah. All right, I'll move you guys over so I can see you a little better. Okay, so fresh and saltwater fishes of the Rio Grande Valley. I'm just going to give you a brief primer on the different groups that you might find down here. Of course, there will be others that I don't cover, but I'm covering the major groups today. So in my lab, I use applied ecology and various technology, many times off-the-shelf technology to answer scientific questions. So I've got a number of ongoing research projects in my lab, including investigation of threatened amphibians, the black-spotted newt, and the lesser siren using environmental DNA. So we actually can sample these critters and their presence just using a cup full of water and extract the DNA out and actually look to see if they're present or not. Also some road ecology work with animal underpasses and wildlife crossings on SH-100 and also FM-106, as Robin mentioned earlier. Snook acoustic telemetry. So we've been following snook around in the Bahia Grande using these little acoustic pingers to look at their movements back and forth, and it turns out that quite a few of them go in and out of the Bahia Grande a lot. Side scan sonar mapping of artificial reefs. So we look at the longevity of artificial reef materials. And in fact, an interesting confluence of materials that happened was from the SH-100 project where they were rebuilding the roads. Those box culverts that were 10 feet by 10 feet ended up in the artificial reef. They were deployed out there and now make excellent fish habitat. And also using time-lapse video to monitor the occurrence of these really small fishes on low-profile artificial reefs. Look and see if we're actually generating new fish on these reefs as the reef matures. So I'm with the School of Earth Environmental Marine Sciences, and we have a couple of campuses, one in Port Isabel, where my lab is located. Some of you might have seen it. Pretty nondescript buildings. They're brown. We've got a big metal building, new research booths that we were able to acquire. We've also got 5 portable buildings right next to the swing bridge in Port Isabel, right before you get to Long Island. We've got the research vessel Vollert that we've had for quite some time, and then we've got the educational vessel Ridley Actually, some of the master naturalists have gone out on the path. We hope to start that program up again once COVID and the vaccines take hold. So I'm a marine biologist and a behavioral ecologist. Of course, I love anything to do with fish. I've had a saltwater aquarium since I was about 10 years old. Study fish behavior. I like to also study fish migrations and how humans affect fish and other organisms. And as I've gone through life as a scientist, I realized that we can really mess things up as humans. We have overfished many of our fish species. We damage the habitat. We damage the habitat that the fish need to recruit to grow up so we can capture them as adults and eat them. So any way we can mess things up, we have. We've introduced species from the Pacific into the Gulf of Mexico, such as the lionfish that now are wreaking havoc on many of our reef species. So hopefully we can figure out something to do to mitigate these issues. And so in my PhD research, I studied a small grouper species on oil platforms. I looked at their behavior and looked at their coloration patterns and determined that there's one male that actually controls a harem of females. They display a unique color pattern. They have an intricate social system. If you remove the male, a female can actually turn into a male in about 4.5 weeks. And this is very important because these fish can't move far away from their habitat. They can't just go to another reef 30 miles away. They have to stay where they are. So the largest female turns into a male in this particular case. I also do artificial reef research, and in fact, we have the largest artificial reef placed on the Texas coast. The Rio Grande Bella Reef is 1.2 square miles, and it's about 14 miles up the coast from the jetties. A very interesting reef that's got all different kinds of materials. One of the most famous reefs down here is the Texas Clipper Reef. This was a Liberty ship that then turned into a training ship for Texas A&M. Many students that went to Texas A&M were Sea Aggies that went out and went on cruises on the ship. This is now on the seafloor in about 130 feet of water, about 18 miles offshore. It's a popular diving area because the water can be quite clear for much of the summer. We also have the Port Mansfield Reef, which is 4,000 round culverts and a tugboat, about 7 nautical miles off the coast of Port Mansfield. It's also a very interesting reef. It's been out there for about a decade and actually has a really good snapper fishery. And so with these reefs, there are many questions that we want to answer about them. One is, are they producing fish? Do the fish stay there when we make these reefs? What other fish could be arriving to these reefs that we don't really know about and haven't seen, but might have a chance of occurring there? So if you look in this right-hand corner, upper right-hand corner, you can see this this device here. This is called a SMRF, or a standardized monitoring unit for fisheries research. And it actually is small artificial corals and a mesh around it. We were actually collecting all the little fish that would arrive out of the water column and settle on the reef to determine what types of fish could recruit. Down below, we advanced to putting out actually concrete structures such as cinder blocks to see if we could improve the recruitment of red snapper specifically to actually seed the reef with new juveniles. Over here on the lower right-hand corner, you'll see an acoustic receiver, and we were very happy because this was February and the water is about 51 degrees, and we located the last receiver that we had used to track red snapper on this reef to see how they were functioning. One of my recent graduate students, Kirsten Laura, who's now at Texas Parks and Wildlife, did some work with time-lapse video cameras to look and see what types of fish and other animals were actually occurring on these low-profile reef modules, such as cinder blocks and some custom engineered ones we have, and even the background that I have, these limestone blocks that we deployed in mounds to see how they function. And she found that these deployments actually even supported Kemp's Ridley sea turtles. They were actually feeding on the reef and actually frequenting them, and unfortunately biting our lenses of our cameras occasionally where we'd have to go buy another camera after they had destroyed it. So tagging red snapper is a challenging effort, especially on these deep reefs. One of my earliest students, Andres Garcia, who's now a biologist at TPWD, used little kiddie poles, like a little SpongeBob kiddie pole, and to go down on the reef and actually catch red snapper that we would then tag underwater. We would put acoustic telemetry tags that would ping their identification and their depth every 2 minutes. And you can see here with the reference of the quarter, this is how big the tag was. We'd actually do surgery and suturing underwater with these fish to implant the tags, and also put a marker tag that if a fisherman caught the fish, they could actually call back and tell us about it. And with snook research, I've been doing snook research since before I came down to the valley. We first came down to actually capture them, to bring them back for a spawning program in Port Aransas. We were trying to see if we could spawn them in captivity and maybe produce more fish for the fishery. That research is still ongoing up there. But what I've done since is actually look to see how the snook are actually utilizing the areas down here. And since we have 2 species of snook that overlap in their sizes and their distributions, we wanted to see if they needed to be managed as separate species. That work is ongoing. Currently, Texas Parks and Wildlife manages all the snook as one species, so the limits apply to both of them indiscriminately. And one fish never reaches the size limit to take them from the fishery. They don't reach the 24 inches that you need to have to harvest them. So one fish gets to survive, never gets taken by fishermen. The other species gets taken right at the size when they're changing sex. So we're doing some research to look at where they feed, where they stay most of the time in these habitats along the lower Laguna Madre, to see if maybe we need to make some changes in their management. And so now we get to the part of the presentation where I'm going to tell you all I know about the local fish, especially the fish families, and how you would find out how to identify fish that you might encounter down here. So if you have a fish in your hand, or a friend sends a picture of a fish and they want you as a Texas master naturalist to identify it, what could you do? You could look at websites, you could contact local experts like me or someone at a museum to help you with it. You could buy some fish keys or get some fish keys online and try to key the things out yourself, which we'll cover in a minute. And the best way to do this is probably with local fish keys or annotated lists. So these are checklists that experts have developed to document every species that they have come across in an area. And so wherever you go in the world, you might be able to find an annotated list to use. If you move to a new area or a new country sometime in your life, you could look up and try to find an annotated list of the species that occur around you. And this doesn't go just for fish. You can do this for mammals and amphibians and insects even. And one of the first things to do is to rule out fish species grouped by habitat, body size, and geographic range. So if you find a fish in India, you probably don't want to use a fish key or identify fish that occur in the United States. You also, if you catch a fish in a freshwater lake or even a spring-fed lake, you probably can rule out any of the saltwater species that you might encounter. And also, some fish are more likely to be encountered near shore versus offshore. And today I'm only going to be covering mainly near shore species. I'm not going to cover any of the tunas and other things that we might encounter offshore. And body size, if you get a fish that's 4 feet long, you can probably exclude many fish that such as cyprinids, such as minnows and things like that, that may not get that big. And geographic range can apply many times. So if you're in South Texas, some fish that occur in northern lakes may be able to be excluded. However, this is something I would caution about, because that's only sometimes. Fish can be moved by humans, of course, and transplanted in other areas, or they may be stocked for a fishery purpose. So you have to be mindful of that. And so we have fish classification schemes in science that we use to categorize all species. And the classification scheme, at least at the lower part of it, is class, order, family, genus, species. Species is the last step, and there can only be one of these. So scientists take great efforts to make sure that there's only one type of organism that is identified as that species, and it only has one name. And then actually, I guess I misspoke there. It has two names. It has a genus and a species, but it can only have the specific epithet, which is the last part of its name, the species name. And so common names are what we oftentimes encounter when we're talking to local people, or we're on a fishing boat, or you have a fisherman next to you and you catch something. They're going to tell you the common name, not the scientific name. And these names can differ by region and sometimes even by person you talk to. Several species may be called the same name. And examples of this might be snook, which there can be several of, perch, drum, and I've encountered several times black grouper mislabeled as other things. And that's why I say scientific name, you can only have one that has this specific genus and species name. However, when it comes to common name, there can be 3 or 4 that have this same common name. And they vary by region. So some examples of this could be redfish. So here we have a redfish that we probably all like to catch along the coastline. That's the red drum or channel bass in some areas. Redfish can also indicate red snapper, which is not a red drum. And another example, spotted trout or spotted sea trout can be a speckled trout or a spotted squeegee if you're in an area near Louisiana. So scientific name is the most specific and the most authoritative. An example of this is the spotted sea trout, which is Cynocium nebulosus. No other organism should have this name scientifically, and there are a list of specific features that define this name scientifically that you can look up. Now, to look these things up, you need fish identification resources. One good one for saltwater fishes is The Fishes of the Gulf of Mexico by Hosey and Moore. You can find this thing used sometimes for $5. I'll have to caution you that the pictures in this book are sometimes not very good. The key itself is very good. The features that you need to identify a specific organism are very good. But I think it was Moore's son who went around when they were trying to publish this book. They didn't have some of the pictures they needed, so they even went to taxidermy areas and they went to restaurants and took pictures of fish on the wall in order to finalize this book. However, where the pictures lack, the key is very good. There's a fish key called the Murdy key, which is now updated but can't be published. It is also a very good key, and it doesn't have pictures, but it has drawn silhouettes, and key features are highlighted by the artist. And actually, I can provide a key, the updated key, for free to you guys, and I can give that to you, and you as a PDF, so you can have it. The Fishes of the Gulf of Mexico, or Fishes of Atlantic and Gulf of Mexico by Robbins. So some of these Robbins keys are also very good. The Freshwater Fishes key is also very good. You can purchase these online as well from Amazon or as a used copy. I find with these Robbins keys that the previous version is not much different than the new version, and you can get them for a lot cheaper. You can also find these for $5 or $6, even sometimes not used, but an older version. Electronic resources, of course, are very valuable these days. Fishbase and an internet search can be useful. Although when you're looking at things on the internet, I have a rule of 3 to follow. So I have to find confirmation from 3 independent sites before I'm confident I found what I thought I found. And that's because if you do some digging on the internet, you'll find that these are circular references a lot of times. The picture you find in one database is the picture that's linked to someone else. And if the first person misidentified it, it's misidentified 3 times. So here's one good resource, the Freshwater Fishes of Texas, and I'll show you how these work here in just a second. I'm going to go through 3 of these, or actually 4, and then I'll go to the links and show you how they work. So this is actually by the University of Texas at Austin. The museum there, they actually have a virtual museum, and they have all the resources and database in one place for many of the fishes. The Texas Marine Species ID is provided by Texas A&M Galveston, and it is an interesting database that has fishes, crabs, shrimps, mollusks, miscellaneous invertebrates, vegetation, and even shells. So you can identify seashells many times in the mollusk section using this particular species ID website. And so, let me see, let me switch to the databases I wanted to show you. Okay, see the Wikipedia article I'm looking at? Okay, so Wikipedia has actually come a long way in terms of what they have for information about fish, and I'm very impressed because a lot of scientists help curate these. And they have, for instance, this is the red drum, a very common species down here. People like to capture Cyanops ocellatus. They have the common names. They have a lot of different places you can look for pictures. They have a characteristic picture here. They give you a list of the characteristics, what you might find. You know, you might find a 3-year-old red drum weighs about 6 to 8 pounds. Record drum's 94, so they have a lot of trivia about them. Their distributions. They talk about their reproduction and growth. We know a lot about that because we actually can spawn these fish in captivity and study them in a controlled environment. Consumption, these fish almost went commercially extinct because a Louisiana chef actually found a special ingredient with his Cajun seasoning and made black and red fish, and then there was a big craze for black and red fish, and they gill netted them until you almost couldn't catch them anymore. Relationship to humans, they also have many references you can look up on Wikipedia articles about where they found the information, so they've got good citations as well. The other thing I like is that for scientific classification, they show you the binomial name, Cyanops ocellatus, and who identified it, which is Carolus Linnaeus in 1766. They also show you the family, so you can actually click on the family name, and it tells you why they're in this particular family group. They include the drums and croakers. They make a repetitive throbbing or drumming sound. They all have this characteristic. The family consists of about, between 280 and 290 species, and it gives you examples of what those species might be. So this is a very good database these days for using for identifying fish. So here is the Fishes of Texas database that I talked about. So this is at the University of Texas at Austin, but it includes Texas Parks and Wildlife and many other collaborators as well. And the thing I like about this database is that you can go to a key of the species, and this is actually a digital key, version of a key that I oftentimes recommend to the Texas master naturalists. But it includes all of these families to search through. It gives you the, for instance, if we wanted to look up, say, a Texas cichlid or a tilapia, it actually has keys to the cichlids, and if you, well, that one has page not found. That's not good. But say, for instance, the blue tilapia, Oricromis mozambicus, you can actually look at the records from the tilapia over here on the right-hand side and see when the first records were posted and where you might find these things. So in the case of where you might find these, this is a very good resource because you can see these suspect, verified, and unverified citations of these fish here. You've got a description, etymology, so if you want to know why it's called Orchromis mozambicus, what the specific characters are, how big it might get. This one gets 360 millimeters, which is just over a foot long, and how you might key it out. So it explains why there's a Texas distribution and why they might be here. So it's got some really good information about each fish species as well. They also have on the checklists. So if you go to the checklist, you can actually sort the data in this database by river basin. So, or a sub-basin even. So let's look at sub-basin. So if we would like to look at, see if the Rio Grande is in here. So you can see all these different sub-basins that you can actually look. Austin, Travis Lakes and Austin, San Marcos, Mission Aransas, Nueces up north, Corpus Christi Bay, Rio Grande at Fort Quitman, Rio Grande Amistad, Rio Grande closed basins, Rio Grande, say if we went to Rio Grande at Falcon River or the lower Rio Grande here, we can actually click the lower Rio Grande as a basin or a sub-basin, and it will compile all of the fish that were within that sub-basin. And it's going a little slow, so I will probably click off of this. I'll go to FishBase. Maybe this will update by then. So FishBase is another website. It's www.fishbase.org, but actually they have several different websites. Fishbase.org is down for some reason right now, so we're going to here's fishbase.us. But there's so many different sites. They all contain the same information and have numerous languages that you can look things up in. But you can search by common name, and you can search by genus and species or genus plus species. So if I wanted to say, well, I think I have a seahorse, we can put that in there. And we can look up all of these areas that a seahorse might be. You can see we've got Cuba, Malaysia, the Azores, UK, Puerto Rico. So if we wanted to go look at a seahorse, Hippocampus erectus, we can actually go to the species itself. It will have a picture of the species. The great thing about FishBase is it actually has a map with the distribution of this species that's known to science. And you can see that it covers our area over here. And they've got multiple pictures. They even have links to Google Images, so you can find other pictures. It gives you the entire classification of the species and the etymology. Tells you where the distribution is, tells you what size you might find them in. It tells you how many dorsal fin spines and dorsal soft rays it might have, biology. And then down at the bottom, that's why I say this is kind of the most authoritative and most extensive. This is like the sledgehammer for identifying species, because you can look up all of these aspects of this particular species, such as age and size, growth, genetics, what their larvae look like. So if you want to go to the larvae, there's some information that's been submitted about the larvae and what the length at birth is. So there's 9 millimeters to 11 millimeters. Place of development and close association with substrate, typically coral reef, things like that. So lots and lots of information there. They even have, in some cases, they have a sound that you can actually look up, and it will actually make the sound, or you can download the sound file, because they actually make some noises that have been documented. Okay, let's see if that checklist. Okay, So now, that was FishBase, and FishBase is a really good resource. You can search by common name or scientific name. As I said, this is the most authoritative. It's curated by scientists all over the world and updated frequently. And so if you found something on the web and you think you have the right ID, you could come here and use this to really help make sure that you have what you think you have. There are also keys, fish keys here as well that you can use. Okay, let's go back to the fishes of Texas. So now I selected sub-basin Lower Rio Grande, and now there are 118 species. Okay, someone asked the question, why do fish make sounds? Well, water is actually a very good medium for sound because it travels so far. They make sounds to communicate with others, mainly to communicate, and sometimes as a deterrent for predation. But almost always it's for communication with their same species, especially for mating. So we can see here 118 species in the Lower Rio Grande Valley area. And as I'm going to show you today, the families are listed, so you can actually look at the categorization by family, and then there are several species within each family group. Okay, and they also list on this database, are these native or non-native? And you can clearly see 3 non-native species here. Non-native cichlid. Let's see if there are any other non-natives. Okay, temperate basses, white bass or sand bass as they're called in some other areas, introduced. Fantail mullet. So these are all things that have been documented here as species that maybe shouldn't be here. Okay, and within here, say for instance, if we went to the striped mullet, a very common species around here in fresh and saltwater, you can look at the documented species all the way from the 1870s. And the gray dots are actually all of the species in the database. So those are kind of a nuisance, but kind of see when you're into the 2010s, look at all the places these have actually been, well, they've been cited, but maybe unverified. But you can see they're all along the coastline here and maybe somewhat inshore, all the way to up in some of these river basins. Okay. So that is the fishes of Texas. And now the final database I'll show you is the Texas Marine Species Identification Database. Their pictures aren't the greatest, but they also have lists of other organisms that you may want to know about, like crabs, shrimps, and even mollusks and other invertebrates. So if we went to the mollusks, you can actually look, they've got information about anatomy. You can look at the different pictures as you scroll down to see what shell you have in your hand. If you're on the beach and you want to know what you have, you could look these up. So very good resource. Let's go back to the fish. And for instance, here, let's just start with greater amberjack. Okay, they've got a picture of a greater amberjack. They've got different details to help key it out. Different pictures showing where those distinguishing features are. It tells you the good thing about one is a similar species that actually might help you key this out. And where you might find them, what kind of habitat, Gulf of Mexico, open waters, near the bottom, sometimes in schools. Maximum size, 6 feet, common to 3.5 feet. So some good information in here about the greater amberjack. All right, so those are the keys that I will share with you. So now I'm going to share my presentation again. let's see here. All right, can everyone see? Let's see if we can see this again. All right, can everyone see see this one again?
Oh, yes, sir.
Okay, thank you. Sorry, I'll make sure I don't move ahead before. Okay, so now let's dig in a little bit into characteristics of fish and where they live. So we have freshwater and saltwater fish and everything in between. But what's the difference between living in freshwater and saltwater? Physiologically, quite a bit. Freshwater fish must retain as many minerals as possible so they're not diluted by the fresh water. Saltwater fish, in turn, must drink and urinate almost constantly to flush out the waste products and gain enough water from their surrounding environment. So we have names for these types of fish. Euryhaline fish can withstand a wide variety of salinities. And examples of these types of fish are coastal fish, fish that occur in most bays. Stenohaline fish, on the other hand, must live in a very narrow range of salinities. So fish that are impounded by water bodies, such as ones that live maybe up in mountain streams and things like that, are not very tolerant to any salinity. They have to live in fresh water. Most open ocean fish aren't tolerant for fresh water and can only stand full strength seawater. And so if they come to if you bring them in and put them in lower strength seawater, some fish may die. The freshwater fish are most susceptible to this. So if you, a lot of times, freshwater fish that come from very far inland cannot handle any changes in salinity. Now, fish and other groups are oftentimes called cold-blooded because they don't really regulate their body temperature. However, the correct term for this is poikilothermic, because that is defined as having a body temperature that varies with the temperature of the surroundings. And they can behaviorally manipulate this by actually moving to other surroundings that might be warmer or cooler. They can go in the shade in some cases, or go deeper in the water and modify their body temperatures. Oftentimes when it's very cold, like we're having right now, you'll see fish, when the sun comes up, you'll see them high up at the surface, and they're actually trying to sun themselves a little bit, try to warm up rather than being in that cold layer of water. The upper layer of water will be warmer. Metabolic requirements go up with temperature, but hopefully not too much. So fish that are cold water species have a really hard time when the water warms up, and we have a problem with global climate change, where these fish that are adapted for a cooler water environment won't have any place to go because they don't have the metabolic machinery to modify their metabolism. So their metabolism will go up really high and almost burn out like a match because they can't control their metabolism. So why is water quality important? Fish perform all of their bodily functions in water. They eat, sleep, breathe, excrete waste, reproduce, and take in and lose salts all in the same water body. And so if it's a polluted water body, or one that has a lot of toxins in it, or even their own toxins, such as ammonia, that can really mess up their metabolic machinery and their physiology, cause them to die. So fish can withstand extreme conditions such as low dissolved oxygen, high temperatures, extreme toxin levels, pure water, all the way to 100 parts per thousand seawater, which is very, very salty. And I've got an example for you to show you some of the extremes that they can handle. So a student that was up at the University of Texas Marine Science Institute near Port Aransas conducted a study on juvenile red drum to see where they might go and what types of environments they might encounter. And so Rafa Perez used data sondes and collected data every 30 minutes for dissolved oxygen, pH, salinity, and temperature. And he conducted these at 3 locations: the inlet to the jetties, a seagrass bed 1 and seagrass bed 2. And this is the data that he collected and over time, right? So Here we have the inlet, air, and the seagrass beds are in the black line in this top temperature one. You can see the temperature varies up and down throughout the days as we're going. When the cold front comes through, you can see the water, well, the air temperature especially goes way down, but the water temperature is slightly buffered. Now, if we look at the dissolved oxygen levels and look at the inlet, it's pretty constant in terms of the dissolved oxygen. However, the seagrass bed site actually varies up and down quite a bit. You can also see a commensurate change in the pH up and down. But what I'd like you to notice here is that in terms of dissolved oxygen, we're getting down to below hypoxic levels, which are unsustainable for many fish species. And this is in a seagrass bed, in an area where you think, I don't know, during the day, you can watch the seagrass leaves actually bubble oxygen off of them, right? But somehow at night, we're seeing these really big drops in dissolved oxygen. Can anyone suggest why we might be seeing these big drops in dissolved oxygen in the seagrass bed? Colder, good guess, but no. Well, photosynthesis has one part of it.
Current carrying the oxygen away.
Yeah, so plants actually produce a lot of oxygen, right? They're responsible for oxygenating the earth. However, seagrasses do what all plants do at night, which is actually consume oxygen while they're making or actually using their sugars and making more of themselves, right? Physiological processes and growth and things like that. They make energy during the day and produce oxygen, but at night they metabolize just like other animals do. And so they're actually in direct competition with everything else living in the seagrass bed at night for oxygen. And so you can see that this is the case because the pH also drops. So carbon dioxide is being produced and oxygen is being used at night. And for large fish, they can't stay in these seagrass beds at nighttime. They have to leave. And so the juvenile fish actually are adapted for surviving in even lower oxygen environments and can actually handle living here and not being eaten by their bigger counterparts. Okay, another interesting thing about fish, especially in the marine environment, is that most fish start out as a fertilized egg floating in the water column at less than 1 millimeter in diameter. And now I want you to see over here on the right, these are actually blue marlin up here at the top and sailfish at the bottom. This is from a colleague of mine at Texas A&M Galveston who studies larval marlin. But this is a marlin that will end up being, 800 pounds later in its life if it lives that long. They look and function very similarly to their adult counterparts. The sailfish in particular actually raise their sails and hunt after little fish in the lights. If you see them way offshore, they look like just a miniaturized sailfish. But they all start out and develop from a fertilized egg floating in the water column. And that's what most marine species do, no matter what their size. So in terms of fish species worldwide, we have about 34,300 species identified. And in fact, the best place to come up with that number is FishBase, because they have all the catalog fish, and that's the number that they have posted right now. In the Gulf of Mexico, we have about 539 species, with 100 being common. And then the Rio Grande, we have 104 species, but only 20 are common to all parts. So one other interesting thing about the distribution of fish is that saltwater covers about 70% of the surface and freshwater less than 1%. However, marine species encompass about 58% of the fish, whereas freshwater are about 41%, and both that live in fresh and saltwater are about 0.6%. So why do we have almost an even distribution of marine and freshwater species when freshwater only covers about 1% of the Earth's surface? Anybody have a guess about that? So The answer is that it is more diversity in freshwater, but why? And the answer is that these are isolated habitats. So these fish evolved together. They diversified together in these freshwater environments. Whereas in the marine environment, everything is connected for the most part. So since these are, especially in the marine environment, you got these floating eggs. Once a fish spawns and releases all these eggs, they can be distributed hundreds of kilometers away, if not thousands of kilometers sometimes. And so while we have different fish in the Pacific and the Atlantic, in the Atlantic, we have a high connectivity even between the Gulf of Mexico and the Atlantic and even the Southern Atlantic. So that's why we have a lot more species in freshwater compared to the amount of coverage on the Earth. So here's a diagram showing you their distribution as well. So fish have adapted to a wide variety of conditions. I'd like you to consider that in air, we have about 210 milligrams per liter of oxygen, whereas in water, in freshwater, we have a 14 milligrams per liter oxygen. And in saltwater, we have about 8 milligrams per liter oxygen. And that is on a perfect day. That's when we have saturation of oxygen. These can go way down, as I showed you in that Rafa Perez examination of the seagrass beds. They can be down at 2 milligrams per liter. So we're talking 1/100th of the amount of available oxygen that you have in the aerial environment. Now, fish reproduce by external fertilization in general. So they release their gametes, the sperm and the egg, into the water. Difference being in freshwater, fish eggs stick to the bottom and stay on the bottom, and the fish hatch at a fast rate and they develop very quickly, oftentimes being able to eat as soon as they hatch out of the egg. In the marine environment, many fish use a different strategy where they float on the surface and they actually have a prolonged larval stage in development where they have to encounter food or they all die. So they may be floating out 100 miles offshore, floating around in these big egg clusters. And then when they hatch, there has to be food around for them to eat to continue their development. Okay, so now let's talk a little bit about how fish are organized. I told you the classification scheme is class, order, family, genus, species. Most people refer to them by their common names, but we even have an organization for that too. The American Fisheries Society actually publishes official common names for fish, but only scientists really use these. Most lay people have their own common names for the fish that they learn from their parents or other friends. But we do actually have a group that actually designates the common names that we're supposed to be using. Okay, so if you're trying to figure out what kind of fish you have in your hand, fish color is not a good feature by itself. Color fades and changes with depth of the fish, so if you catch it hook and line and you put it in your ice chest, by the time you look at it again, those colorations are probably gone. Fish colors changes with the clarity of the water, so in murky environments or clear environments, you might find different coloration being held by the fish. And the exception to this are dark pigment spots that usually remain present on the fish even after death, such as the ones on this red drum at the bottom, these eye spots, and also this pinfish here that has a dark melanophore right next to its operculum. So here is the fish classification scheme I was talking about, and class. So if we're talking about a red drum here, class would be Actinopterygii, which is the ray fin fishes. It's in the order Perciformes or perch-like fishes. The family Cyanidae, or the drums, which incorporate the redfish, croaker, spotted sea trout, sand sea trout, whiting, and other things, all have this drumming capability. The genus Cyanops and the species Ocellatus. Now, each one of these names actually has some meaning to it. Some of them can be names of famous scientists. In this case, Ocellatus has to do with these spots or eye spots that occur on its body. Okay, here's a slide showing you different measurements we might make on a fish for fishing or for scientific purposes. The common measurement you might encounter would be the total length of the fish or the fork length of the fish. And that would be from the tip of the jaw all the way to the maximum length of the tail. A fork length would be the center portion here. So a fish with a highly forked tail would be measured towards the center. But we have all different other kinds of measurements that we can make. The length of the spines, the length of the rays, the standard length, which actually incorporates all the vertebral column. We've got the girth, which is the circumference of the fish. That might give us an idea of their health. We've got any number of measurements that we can make. We also count the number of scales that actually incorporate the lateral line or the sensory apparatus on the side of the fish, because those can be useful for identifying specific species and things like that. But the most common length that you would encounter would be the total length, which would be a fish laying flat on a surface from the tip of the jaw all the way to the maximum length of the tail. Okay, for identifying fishes, we use what's called a dichotomous key. And a dichotomous key is a series of couplets or paired questions that help to identify groups of organisms. And some of these uses are, of course, to identify fish to species, and also to identify and emphasize recognizable difference between the species to do that. The drawbacks of dichotomous keys are they're time consuming because you have to go through each step to finally figure out what you have. And they are made by humans and therefore flawed because it was the person's idea who made the key to decide what features you should look at. And also, species traits can be variable and exact statements rarely apply to all fish. So that's another thing to remember when using keys. That's why I always recommend trying to use more than one to arrive at your answer and to verify that you arrived at the right answer. So here's an example of a very simple key. So one that everyone should know, these are vehicles familiar to everyone. So if we start with the first couplet or the first two questions, you start at the top, you say propelled by internal combustion engine. Okay, if I'm thinking of a tricycle, then I would answer no to the first question and go to the second question, which says propelled by muscular effort of the rider. Go to item 3. When you arrive at item 3, you go down to step 3. It says vehicle with 3 wheels or vehicle with 2 wheels. You would select vehicle with 3 wheels. Your answer would be tricycle. It is a very simple example, but on a fish key, you would arrive at the genus and species of the fish that you were trying to identify. Okay, here's another key. We're keying out seahorses. And this is actually from that Murdy key that I can provide to you, which I think will be useful. Now, we've got, I'm starting in the middle here. We've already arrived at, we think we have a seahorse. But if we went to question 1, we would say tail prehensile, caudal fin absent. Or the second question's tail straight, not prehensile, caudal fin present. And that would be the tail fin. So in this case, if we're thinking about a seahorse, the caudal fin or the fin at the end is not there because it has a curved tail. So we would proceed to step 2, which would be first dorsal fin rays 10 to 13, pectoral fin rays 10 to 13, and tail rings 28 to 34. So you might have to get pretty specific to identify these things, but you could count the tail rings and look and see, oh, if I find one that's 28 to 34, it's a dwarf seahorse. If I find one that has 33 to 38, it is a line seahorse. Okay, so you could actually use these features, and there's a glossary in the key as well to actually help you identify what dorsal fin rays are and what pectoral fin rays are. So you could actually try to key out your own fish this way. Okay, and now what I like to do in this course is just give you an introduction to the description of fish families that you might encounter in South Texas. That's because there are so many species down here, we can't cover them all. But if I tell you kind of the key traits that you might find in these particular fish families, you can go to a fish key and quickly identify what you have if you know what family to look under. Because that's a higher categorization scheme. There may be 10 species underneath that family or 20 species, but you don't have to look through all 500 to find what you're trying to find. Okay, and this only encompasses fish that occur near the Rio Grande, near Brazos Santiago Pass, in the fresh or the coastal environments. So family Dasyatidae are the Whiptails stingrays, and one of the most characteristic species we have of that is the southern stingray. It's very large. It can be up to 3 feet across on its wings here. But one thing about stingrays is they always have a whiptail and a stinger. And the stinger is on the base of the tail. And these are fish that you might encounter anywhere in the coastal environment. And I like to suggest that you walk a certain way when you're in a coastal environment called the stingray shuffle. And you will avoid ever being stung by a stingray if you do this. And that is to walk never by stepping and taking big steps, but actually shuffling your feet in the sand. Because the biggest predator of stingrays are sharks. And the only defense that the stingray has is its stinger or to swim away. If it feels pressure on the part of its body like it's being pushed down or bitten, it will try to strike with the stinger. However, if you shuffle and pick it up from its underside, it will simply swim away. And so if you're ever wade fishing and things like that, try not to step in big steps, but actually shuffle your feet, and you will avoid being stung by one of these many, many stingrays that we have in coastal Gulf of Mexico. Okay, another big fish group that we might find are the Lepisosteidae or the gars. These are commonly found in freshwater environments. However, they can also move to saltwater environments, especially after heavy rains. One of the largest ones we have, here's river monster guy, actually with an alligator gar. Alligator gar are common in the resacas around here. They're sold as food called katan and fried in many seafood restaurants. The other species we have here is a spotted gar. It doesn't get as big. It only gets about 2 feet long. But they both have a mouth full of teeth, and they prey on small fishes for the most part. So the next family we have is a Lopidae, and these are also called 10-pounders. And we have two groups that are very similar. We have a Lopidae, and then we have Mughulidae. Mughulidae are the tarpons, and Lopidae are the 10-pounders or ladyfish. Both these fish groups fight very hard in the when they're hooked, and they're often targeted by fishermen because of that. Tarpons, in particular, are very large, and actually can live to be about 80 years old. Someone's asking, why are Lopidae called 10-pounders? Well, it's because these fish only get a few pounds, but they fight like they weigh 10 pounds. So this group is known for their heavy fighting and targeting by recreational fishermen. But tarpons can live to be 80 years old and get about 6 feet long. And in fact, Fort Isabel at one point was called Tarpon, Texas, because of so many tarpon being captured every season. However, that's not the case anymore because we've dammed up most of our rivers, and we've also over-harvested tarpon throughout the last 50 years. Okay, another species that can frequent both the freshwater and marine environments is family Mughulidae, which are the freshwater eels. They're actually catatomous. They go back to saltwater to spawn. They spawn in the very deep ocean, but if you were to encounter a really black, slimy eel, and in fact, I actually saw a picture from a student of mine the other day, and it was kind of greenish colored, but they produce a lot of slime, and they look like a characteristic eel. And these do occur along the coast here. They're a big food fish along the eastern seaboard, and of course in Europe as well. They spawn in the middle of the Sargasso Sea, way offshore. They swim all the way out there, spawn at really great depths, and then their larvae actually make it all the way back to freshwater inlets all along the Gulf and the Atlantic seaboard. And then we have some other eels. So these are marine eels in family Ichthyology. They're called snake eels because they look snake-like, and they also have distinctive coloration patterns. But you typically find these on natural reefs and even in coral reefs. But occasionally one will be captured by hook and line if someone's accidentally fishing over a nearby natural reef. Then family Mughulidae, which are anchovies. Anchovies are a fish that you'll find in huge numbers at some times of the year. They're a pretty nondescript fish that has silver. You can actually see their air bladder through their translucent skin. And you can also find a big silver line down the side of their body. There are two groups that you might find that might have these characteristics of being somewhat translucent and silvery and have a bright silver stripe down the side. I'll get to the other in a minute, but for anchovies, you want to know if you have family Mughulidae or an anchovy in your hand. If you push the snout of this fish, it actually has a bulbous gelatinous nose. So you can actually, it's quite flexible, and you can push it and tell that you have an anchovy versus one of these Monidia that we'll get to in a minute. Now, family Cyprinidae is another silvery fish, but you'll only find these in the freshwater environment, whereas the anchovies are saltwater. These are throughout North America. There are silvery fish. If you ever go fishing in lakes and you buy minnows, these are also cyprinids. Cyprinids can be larger, such as smallmouth buffalo and other large fish with scales. Somebody was asking a question there. It popped up and now disappeared. Let's see what this is. Yeah, so the anchovy actually will retain its gelatinous nose after preservation, so you can actually use that as a feature to look at it. If you keep it in an ice chest or even preserve it, it will still be gelatinous. But family Cyprinidae, there are about 2,700 species throughout the world, and we have quite a few of them in North America. And in fact, they're the most abundant freshwater group that we have in North American freshwater bodies. One poster child in the Cyprinidae that we have in the Rio Grande is the Rio Grande silvery minnow. It's actually a threatened species federally, and it doesn't make it past Falcon Dam because we've dammed up the rivers. And this particular species actually has a pelagic reproductive state. So it actually fertilizes the eggs and has floating eggs that have to float down the river. They hatch and they swim back up the river to habitats. But since we've dammed the rivers, there's no place for these to go. They all die when the eggs make it past Falcon Dam. The other problem is that we have really salty water as we get further and further down the river. And these fish are only adapted for freshwater. We messed it up so bad that in New Mexico, the Rio Grande silvery minnow is actually removed from the river after the snow melt happens, and they're put in impoundments off to the side of the river, and people have to take care of them all year long. They put these fish back in the river once the waters come back again. So it's pretty extreme example of what we've done to mess up our freshwater habitats. But family Cyprinidae, often silvery scales on the side. A silvery fish that you might find in freshwater is oftentimes family Cyprinidae. We have one relative of the piranha here in South Texas. Family Characidae are the tetras and the piranhas. We have one called the Mexican tetra that actually occurs all the way up through about San Marcos now. They've been introduced further up and taken far and wide. But actually, if you open up the mouth of a silvery fish and you see something that looks like piranha teeth, and it's in fresh water, you likely have a characid. The characids are very, very abundant in Central and South America, but we only have one example here. So in freshwater, we have several catfish species, and these are in family Ichthyology. Bullhead catfishes, such as channel cats and things like that, are very common and occur in many of our rivers and lakes. We also have saltwater catfishes, but they are in a different family. So if you catch one in freshwater, it's in one family, but in saltwater we have family Ariidae, which is composed of two species: the gaff-top sail catfish, which is the one here that actually has a really long extra spine on top, and also has a fin ray and a fin ray that comes out of the pectoral fin as well. So they've got these extra tabs that stick out. And the hardhead catfish, which looks similar but does not have these extra frilly ends on the dorsal fin and the pectoral fins. This one is, most fishermen say, is good to eat. The hardhead catfish, most people say throw it back because it's got a lot of red muscle tissue. And we also have an Armadillo de Rio. We have family Lopidae, which are introduced suckermouth catfishes. Now we have two species that occur in many of the resacas in the Rio Grande down here, and in fact in other warm freshwater bodies throughout the United States now. These are becoming more common. They tear up the banks because they, when they lay their eggs, they dig into the side of the bank, and you can lose considerable amount of real estate by these guys collapsing banks around resacas and other lakes and things like that. It's supposed that these were released from aquarium fish, from people as pets, having them outgrow their aquarium and then putting them in the lake because they didn't want to kill them. And now any resaca you go to, if it's got a walkway or you can walk up to the bank, you can probably see one of these sucking and cleaning off a piece of wood or a rock or something like that, because they really like to eat invertebrates and algae. And believe it or not, we actually had a salmon here, or a salmonid in South Texas. We had the rainbow trout introduced as a put and take fishery in many of the water bodies down here. This is especially true because winter Texans like to come down and catch rainbow trout that they're used to up north. We actually still have on the books a license and a tag for catching rainbow trout in South Texas. However, this is a ridiculous idea because these fish cannot handle very high temperature water, and so they would only survive in the winter time. Once the water got about mid 70s or upper 70s, these fish would die, and they would have to restock them the next year. So family Salmonidae, the salmons or trouts, we have one representative in some of the lakes in Texas, but down here is only introduced and only would survive in the wintertime. So family Mughulidae, or the mullets, are also a very common species in fresh and saltwater. And in fact, in many of the resacas, you can find mullets as well because they've been pumped in through the water intake pumps. These are euryhaline, which means they can withstand either fresh and saltwater. This is the fish group that thinks it's a bird because they have a gizzard for grinding up the algae that they eat off of rocks and other areas. They have somewhat of a beak. Their lips look like a beak, and they also try to fly, because if you've ever seen these, even in the surf, you'll see them jumping out of the water. Sometimes it looks like they're just having fun, but oftentimes it's because there's predators behind them. So this is a bait fish. This is also a food fish for many predatory fish as well. So someone was asking, are they considered invasive, and we're trying to get rid of them? Is that the Lopidae or the suckermouth catfishes? I would say no, because they're in all the resacas and they're in super high abundance. You're supposed to kill them if you catch them, but people are not actively trying to get them out of the resacas. And in fact, a funny story I have is that we were watching the news one day, and this woman was very concerned because all of her favorite fish were going to die in this resaca because they were pumping it out to dredge it and clean it up. And wouldn't you know it, the fish that she was worried about and they transferred into another resaca nearby were suckermouth catfishes and tilapia. So these were two introduced species and other things. And so we were like, why are you saving those? Why would you do that? And they don't know any better, right? Because they're the fish we have in the resacas now. Okay, and this is the other silvery fish group that you might encounter actually in both fresh and saltwater environments. You'll find them really near the, I guess, the surf zone. You'll find them in the bay. Find them in lots of places, and they also have a really bright silver stripe going down the side. But these are in family Ephemeropsidae. These are the New World silver sides, but they do not have that bumper nose like the anchovy has. They actually have a very hard snout, but they do have very similar characteristics. They're somewhat translucent. They have this bright silver stripe. These are actually introduced into many freshwater impoundments as forage fish for largemouth bass and other target species. but they're very translucent with a bright silver stripe on the side. Okay, and I showed you gar a few minutes ago, and now we have another species in saltwater that kind of looks like a gar, and this is family Balonidae, or the needlefishes. They have, lots of teeth in their upper and lower jaw. They mainly live in saltwater, although you can find them in freshwater resaca sometimes. But they just have this big rostrum, big mandible here with little small teeth for eating small fish. And in fact, you can see these often in the lights underneath docks and things like that in the saltwater environment. Say if you're in the lower Laguna Madre, you can see them under the lights chasing little fish and zipping around really fast. They're long and slender, and they've got really big jaws. Okay, now we're on to family Pisciliidae. These are the live bearers. These also occur in fresh and saltwater environments. These actually are called live bearers because they have external fertilization, or actually internal fertilization. The male copulates and fertilizes the female internally. She gestates with the eggs and actually releases live young. These are kind of small, the females are small, almost nondescript fish that you might find, especially in resacas, but the males are very brightly colored and have various ornamentation on their sails. So they have this big ornamentation. They hold harems. They're used a lot for scientific research because of all their complex behaviors and courtship behaviors of females. There's also another fish in this group that's very nondescript called the mosquito fish. If any of you have ever kept like a freshwater pond, oftentimes people will recommend that you keep mosquito fish because they eat mosquito larvae like nobody's business. They are also in this group, but they don't have this ornamentation. They're just a small, almost nondescript fish that you'd find in a freshwater environment. And then we've got another group that's very similar in body shape and size, and this is family Cyprinodontidae. These are the killiefishes and pupfishes. These fish, kind of soft-bodied fish you might catch in the nearshore environment or in a freshwater area. They oftentimes see them feeding at the surface in very low oxygen environments. These fish are very, very much extremophiles. They can live in very, very high salinity environments. So if you find a tide pool or something and you find a fish left in it, it's probably going to be a Cyprinodontidae because there's very few other fish that can survive. And in fact, this family we found in the very back of the Bahia Grande when right after they flooded it. The salinity was 3 times higher than seawater. Almost everything was dead. There were no other fish species, but this family Cyprinodontidae, especially the pupfishes, were in the back, still living and perfectly happy. We often find these fish also in hot springs, in the Devil's River and other areas. You'll find some very, very peculiar species because they can withstand really high temperatures and really high salinities. Okay, another group you'll find both in freshwater and saltwater environments is that family Centropomidae, or the snooks. Okay, as I said, we've got, I think, 3 species here now, maybe 4. The biggest is the common snook. And if you ever catch a fish or see a fish that has a big black line down the side of it, looks like someone took a Sharpie marks a lot and drew a dark line down the side of it, it's most likely a snook. They are very big ambush predators. They're very targeted by fishermen because they fight, I say, like a very largemouth bass, but they get up to 4 feet long. So very, very fun fish to catch. And in fact, in my sampling, I would say they're probably the best tasting marine fish I've ever encountered. And I think that's why they're also overfished in many areas. So Centropomidae, they have kind of this upturned mouth. The lower jaw is longer than the upper one, and they have this big black line down the side of them. Okay, one very common group that we find throughout North America, and we find in many freshwater habitats, are this family Centrarchidae, or the sunfishes and basses. And this includes all the panfishes such as crappie, sunfishes, largemouth and smallmouth basses that you might find in all the freshwater lakes throughout North America. We also have these fish in many of the Resacas and the Rio Grande here. So they have a similar body form. They both have, even the basses and the sunfishes have this dorsal fin. It's kind of connected. It's got spines in the front and soft in the back. Many of the sunfishes have ear spots, or they'll have brightly orange coloration that goes away very quickly once they die. But if you find something that's kind of like a panfish and you find it in the freshwater environment, it's likely a Centrarchidae. And one other fish group that we also find co-occurring with them is family Cichlidae. And we only have one representative that's a native, which are the Rio Grande cichlid. And they have these bright pearlescent spots all over them. They're very beautiful in nature. They actually exhibit parental care where they guard nests of young. So if you find fish that actually have a big bare spot in a shallow water, freshwater environment, and you see fish guarding around them, they may be Rio Grande cichlids. We have a couple of introduced species as well. We have the blue tilapia and the black chin tilapia that are originally from Africa, but they're an aquaculture species that have been introduced to many, many water bodies throughout the lower states of the United States. During Y2K, people thought it would be great to introduce these into every place they could think of because they thought we were going to run out of food. So they're in many spring-fed rivers and things like that up near San Marcos and Austin, all the way throughout Texas. These have been introduced. And they can grow in very high densities. In aquaculture situations, you can grow 1 tilapia per gallon. And so it's, when you're buying these as a food fish in the store, that's about how they've been raised. So you got to consider that when you're eating them. But family Cichlidae, they have more fleshy lips than the Centrarchids do. And the native one has some really, really beautiful pearlescent spots that you'll see. And then another interesting group that you might encounter are the family Gobiidae. These never get very big, but if you're cast netting or netting or picking up rocks, sometimes you'll find these fish still hanging on. And gobies all have this special trait of their pelvic fins are actually fused into a suction cup. And so they will hold onto rocks and other things in fast flowing water. They're oftentimes associated with substrate. You very rarely find them swimming freely. And they're usually very small. They're only a few inches long. The longest one, the longfin goby or highfin goby actually gets maybe 8 inches long, but they're still kind of long, slant, and slender fish with a big head. The naked goby, which has kind of orange with some tiger stripes on it, is the most common small fish that we find in the fresh and marine environments when we're collecting and we count all the species. We almost always find at least a couple of these because they're everywhere that we sample in the Resacas or out in the coastal environment. So someone's asking, how large is the Rio Grande cichlid? The Rio Grande cichlid at maximum can get about 12 to 13 inches long. Oftentimes you'll find them 4 to 5 inches long if you're cast netting in a Resaca or seining, something like that. Okay, another family that you would encounter in a river environment or in saltwater is family Paralictidae, or the sand flounders or left-eyed flounders. These encompass the bay whiff, which is a quite small flounder that you might find and no one would take for a food fish, or the southern flounder, which is one of our highly targeted food fishes along the Texas coast. You'll know these groups from the right-eyed flounders because, of course, the eye migrates to the left-hand side, and so they're always facing up from the left-hand side. Flounders and soles actually have eyes on both sides of their head when they're young, and the eye actually migrates around to one side or the other before they lay down in the sand. And so, very interesting group. If you flip them over, you'll see they're actually a two-sided fish with actually fins on both sides. But they stay flat with their eye facing up once they reach maturity. These fish actually are heavily managed. The southern flounder is targeted a lot when it's about ready to spawn. So people that gig for flounder actually are taking fish that are just about to go reproduce offshore. I know nobody likes to be regulated, but that is one of the smartest regulations they've come up with, is to prevent gigging throughout some part of the spawning season for these flounder, because they all line up to go offshore. The males leave first, and then these very large females that we used to call magic carpets because they could get so big, then leave and meet these groups of males offshore and spawn, and then return back to the bays later. But they, when you gig them, of course, you're harvesting them right when they're full of eggs and right when they're full of sperm, about ready to go make more fish. We have a bad habit of doing that to many fish that we like to eat. Okay, here's the other group of flatfish that you might encounter. These are the American soles. None of these get very large. As you can see, these gloved hand here, this is about maximum size of the sole that we get down here. The line sole and the hog choker are the two species that we have, and they're more rounded than the left-eyed flounders, and also the in this family Anchovies, the eye is almost always on the right-hand side. But as I said, there's always an exception. Sometimes you can find a left-eyed flounder that decided to migrate its eye to the other side, but that's a very rare occurrence. Okay, one fish that you probably won't ever confuse for another family is the puffers in family Tetraodontidae. They're called Tetraodontidae because they actually have split between the upper and lower teeth, so they have basically 4 teeth, and they oftentimes have the protective spines. And when they're not puffed up, they still look like a big block swimming through the water. Very strange looking, very big eyes. But these are oftentimes captured in trawl nets. Sometimes people will catch them hook and line in the nearshore environment, or catch them in a cast net. But they will try to puff up when you capture them. These are in family Tetraodontidae, or the puffers. Okay, one other very common group that you'll find in the coastal environment is family Cyanidae. I've been over this a couple of times when talking about the key examples. But these are the drums. And you've got red drum over here on the left-hand side, spotted sea trout on the right-hand side, Atlantic croaker here on the bottom, and then Gulf kingfish here on the left-hand side. These are all members of the drum species. That's because the males have the capacity to actually drum to communicate with their other of the same species, and they all can have some of these spots on them in various forms. Red drum typically only have one spot near the tail, which is why they're called Cyanops ocellatus. However, these spots can be easily reproduced in the genetic code, just like people can be born with multiple fingers, and these can be all over the body. Okay, the spotted sea trout typically has lots of spots on the top. That's why its species name is nebulosus, or star-like, because it has all these spots on the top. Okay, and if you want to impress your friends, you can actually identify whether you have a male of a drum species or not, because only the males drum. It's actually influenced by hormones circulating through the bloodstream, and when enough testosterone is given, this sonic muscle develops, and this sonic muscle flexes really quickly up against the swim bladder, causing a drumming sound. And it's so loud that in captivity, they'll actually make your feet vibrate in a concrete floor, because it's such low frequency and they drum so much. So I was at a lab where we had big red drum that were spawning in captivity, and at night when I'd be working there, the whole floor would vibrate because they were spawning that night. But if you want to impress your friends, if you feel one drumming, you can say, "Oh, that one's a male," and I know it's a male because it's got a sonic muscle and it's right near the body wall, and you'll find it. So this is in group Cyanidae, the drums. So many of the fish that you might encounter when you catch them hook and line on the Texas coastline will be in this drum family because they're so abundant. Another fish that you'll capture typically by accident is a member of the Speridae, or the porgies. We have 2 representatives here along the Texas coast. that occur in high numbers. One is the pinfish, so named because it feels like someone stuck you with a pin cushion when you grab it, because it's got really, really sharp spines on its dorsal fin and also its anal fin. So it can stick you in your fingers. And they're also hard to hold on to. They're also called piggy perch, because people like to use them as bait. But they all have this characteristic black spot here, and they have yellow and blue lines going down the side of their body. Large ones can get about a foot long, but typically only find 3 or 4 inch fish. Another group of the Asperidae are the sheep's head. And anybody that fishes along the coast knows that seasonally you can catch a whole lot of these, and that's because they are also spawning during that time. That's the only time they'll really bite in high numbers. And so when they're spawning, they get very hungry and they amass in big groups. And that's why you can catch them in high numbers. They're awfully bony, but people love to eat them, and they have a nice white flesh that's very delicious. That's in the Asperidae. And then one of my favorite groups, the Serranidae, which includes the groupers and the sea basses. Fun fact about them is most of these fish change sex sometime in their life. Got all the way from the strawberry grouper or rock hind here to members of this group, which are the Victorperca, which include the cone grouper or the gag, and the scamp, which are highly targeted offshore. And then you've got a really small representative that occurs both inshore and offshore called the belted sandfish. It actually has a really interesting behavior of egg trading, where it actually can perform as a male or a female depending on who it encounters. So these fish actually have both gonads at the same time, and depending on the size of fish it encounters, it can decide it wants to perform as a male or a female on the same day. And so these fish are very plastic when it comes to their reproductive capacity and reproductive organs. They oftentimes have social hierarchies to determine who's male and who's not. And they also are highly targeted as food fish. And so this is in group family Serranidae. And we call this a grab bag because, as you can see in this group, there are big fish that can get, even the Goliath grouper, which is one of the largest representatives, can get 7 feet long, all the way down to this belted sandfish, which may only be 2 or 3 inches long on a rock or a reef area. And another fish group you might find in similar types of habitats as some of these serranids are family Balonidae, or the blennies. You might even catch them in the jetty area. You won't find them in freshwater environments, but you would find them in the jetties and other rocks. But the characteristic part about them that will give them away as blennies is their tufted eyebrows. So they all have these tufts, some very large, some smaller, but they will always have a tuft. And the only other thing you could confuse them with is maybe a goby. But if you look on the underside, you will not see a suction cup formation of their fins on their underside either, or their ventral surface. So they have these rasping teeth, typically big lips, and they have these exquisite eyebrows that you might find right above their eyes. They oftentimes live in little burrows and crevices. Family Cygnathidae is another group that you might encounter. We have 2 representative groups here. We have the pipefishes and the seahorses. The pipefish do not have a prehensile tail. They actually have a tail fin. Of course, the seahorses have a prehensile tail to hold on to everything, and are highly associated with substrate like seagrasses, and you only find them typically in the bay or associated with a reef. They're very interesting, of course, because they have the male actually carries the young around in a pouch, kind of like a kangaroo, and they're responsible for developing the offspring. They are very, very interesting. So if you see a fish with a tube-like structure on the front, a prehensile tail, or even a long tail, but kind of feels like it's got an armor plating on its outside of its body, you probably have something in the family Cygnathidae. Okay, and that's the last family group, and I'd like to give one other plug to a project that I've been heavily associated with, which is the Rio Grande Valley Reef. The Rio Grande Valley Reef is about a 1.2 square mile reefing area. It's the largest area on the Texas coast. And as I said before, here's actually some of these box culverts in this picture that actually came from the State Highway 100 project that are actually now in the reef. There's actually a pile of concrete railroad ties. It's about 32 feet tall in the middle. There are now 4 ships deployed in the reef. And the Friends of the RGV Reef are oftentimes asking for help in terms of boat use or even in terms of donations to help put more reefing materials down. It's just a fantastic reef. And even if you only get a chance to go out there and fish it, there's so many pieces of material that have been deployed so far. There have been about 4 pile or arrangements of material this side that have been deployed on the reef. So huge amount of material. I think now about 20,000 tons of material has been deployed on the bottom of the seafloor in that 1.2 square mile area. And it's the biggest reef on the Texas coast, and it's the jewel of the area down here. So with that, I'd like to thank you for learning some more about fish, and hopefully you'll be able to use some of the information I gave you to key out the next fish and impress your friends about what you know about them.
Thank you, Dr. Klein. Are there any other questions for Dr. Klein before recess?
Yes, I have 2. One's related to the sucker catfish, the armor catfish. What would be the best way to get rid of them? Because personally, I actually have a resaca that has some, so I want to see if I can start getting rid of them. That's the first one. The second one being, what's a good place within South Padre Island and Port Isabel that you can find good clear water that's fairly easy accessible?
Well, in terms of the Lorcardae or the sucker mouth catfishes, I mean, we were successful catching them with gill nets and things like that, but there are so many in the local resacas that I don't know if you'll ever get rid of them. Texas Parks and Wildlife says that if you catch some, you're supposed to kill them and not put them back in the water. But we would catch 100 in a resaca at a time.
Wow.
And so they're everywhere. I don't know what you do. They reproduce heavily. There would have to be an eradication program to do that, but I don't think there's very much political will to do that because the resacas are murky. Probably part of the murkiness has to do with them stirring up the bottom so much and stirring up the sides and all the sediment loss we have. But I don't know. And then you want clear water. Well, clear water is hard to come by a lot of times in the lower Laguna Madre or offshore, and it happens seasonally. So I would say, July, August are your best times to find clear water. But I don't know what kind of depths you're talking about, but if you go 18 miles offshore and what's it, inshore? Yeah,
pretty much inshore, even offshore.
Well, inshore around the seagrass beds, especially the turtle grass beds, maybe south of the convention center, but out into the lower Laguna Madre can be quite clear. You can actually see the individual blades of grass when you're boating over them. Or South Bay, you can find some fairly clear water. But offshore, the Rio Grande Valley Reef is sometimes clear, but further offshore, 18 miles offshore, you can start to get some really clear blue water for many parts of the year. So that's what I would say about that.
Great.
All right, thank you very much.
All right.
Again, Dr. Klein, thank you very much for this evening. And we appreciate your coming and joining us.
All right. Thank you. And, I've got that fish key. Can I send that to one of you? I guess.
Go ahead and send it to me, and Robert and I'll send it out.
Okay, so I'll send you the PDF of it.
Perfect.
Send it out. All right.
Great.
Thank you.
All right. See you later.
I'd like to introduce Tony Reisinger. He has served as the Cameron County Marine Extension Agent for 38 years. He earned a BS degree in zoology from the University of Georgia and a master's degree in biology from the University of Texas in Brownsville. He began his career as a marine biologist for the Georgia Department of Natural Resources and continued as a marine resource specialist with the University of Georgia's Marine Extension Service. Tony moved to Texas to accept the Cameron County position with Texas AgriLife Extension and Texas Seagrass. He's worked with the Gulf of Mexico shrimp industry and has been successfully conducting bycatch reduction compliance training to reduce catches of fish and sea turtles. Reisinger has founded the Texas Red Tide Rangers, a group responsible for determining red tide concentrations and impacts to the South Texas. He founded the Texas Coastal Naturalist Program, a volunteer group responsible for cold stun sea turtle events, marine mammal strandings, and harmful algae blooms. Tony serves as an advisor to the 2 local Texas Master Naturalist chapters, the Rio Grande Valley and the South Texas border chapters, which he helped founded in 2002 and 2015 respectively. He is a published photographer and accomplished artist in Japanese fish printing, and he enjoys teaching that to others. Thank you, Tony, for being with us tonight.
Thank you, Robin.
No problem.
Okay, I've got to figure out how to share my screen. Okay, I'm going to talk to you about ecology today or tonight. And can anybody define ecology for me?
Will this be the study of the environment and the relation it has with the organisms?
You got it. Great job. Who was that? I can't see who answered it. Shadrach?
That was me, Shedek Villafranca.
Okay. Glad you're here too. So we'll start out with the barrier island system in Texas. We have 7 barrier islands along our coast, and they're very important. They play an important role. We also have 6 passes through which water goes in and out into the estuaries. Can anybody define an estuary? Robin says it means marvel of God in Hebrew. Well, that's a great description because they are marvelous places. They're where fresh and salt water mix. And a lot of the fish that Dr. Klein talked about earlier depend on these estuaries to spend their early parts of their lifespan. And it also serves as a nursery for shrimp, or they serve as nurseries for shrimp, which are one of our most important seafood products in the Gulf of Mexico, most valuable seafood product in the Gulf of Mexico. We also have 2 peninsulas that are along the coast. And can anybody name the pass that is on the south end of South Padre Island?
It's me, Shuck, again. That would be the Santiago Brazos.
Right, Brazos Santiago. And does anybody know why Brazos Santiago Pass has that name?
Doesn't it translate to be like the arms of Jesus or something?
You're close. It's the arms of St. James. St. James is the patron saint of Spain, and Santiago is, I guess, his nickname there. And when there were Spanish explorers in the Gulf of Mexico, and they discovered this pass, and they had a hard time out there in the Gulf because it's rough, a lot of waves, a lot of wind, storms. And when they went into there, they felt like when they discovered the pass, they went inshore and they felt like they were in the arms of St. James. So that's why they named it that. I can't remember which explorer named it that, but it is an appropriate name when you compare being in the pass and going and being in the Gulf of Mexico. So our Texas coastline was not always the way we think it, the way it is now. In 13,000 years ago, it was 50 miles offshore. Does anybody know why why the coast was so far out? Has anyone ever heard of sea level rise? Well, sea level rise started about that time and went up to what we are at presently. And we're still experiencing sea level rise, but probably we may be experiencing it at a slower rate right now. Sea level has gone back and forth over the years. So the reason that it was so far out was it was during the ice age, and a lot of the ice in the northern areas of the continent took up, or continents, and it took up a lot of the water that is now back in the oceans. And as you know, we have Greenland melting, we have Antarctica melting. So slowly it's coming back again, going up, going even higher. And I don't know when it's going to stop, but there's been some dire predictions. Maybe not in my lifetime, but some of you younger people may even notice it. I noticed some of the differences that we have going on now. So barrier islands. Padre Island is the longest barrier island in the world. And it's a young island. It formed just several thousand years ago. And it's made up from beaches, dunes, marshes, and tidal flats, which are on the inside of the island, or on the western side of the island. What is the profile of a beach? We're going to start with the Gulf of Mexico, and there are 3 bars in offshore of the Gulf, primarily 3 bars, sandbars that form in the Gulf because of wave action. And then when you go up to the forebeach, you have an area what is called the swash zone. And that's a very important area. And you're going to see some, I guess I'm going to have some examples of some of the animals that live in that area. It's a very dynamic area in the forebeach. And then the back beach is just behind that. And it is also a dynamic place too. Sand dunes begin with what we call the coppice dunes right here, smaller dunes, and anything can start a sand dune. I was looking at, I rode up to the Port Mansfield Cut the other day, and I found one of the classic green bleach bottles that we find on the shore that wash up from Mexico usually. Those were one of the first ones that we started picking up. And there was a small dune forming around it, and there was some even some plant life there. And so we think of plastic as being pretty bad. Well, it does, I guess it did start sand dune in that respect. Then we have the four island dune ridge, and those are some of the larger dunes. We've got some much higher dunes further up the coast between here and the Port Mansfield Cut. The stabilized dunes are the next area, and those have been here for quite some time. The younger ones are the Four Island. And then we go down into what we call the vegetated barrier flat that is comprised of ponds and sometimes marshes and even some lower areas. Underneath that is a cell of fresh water that usually occurs. And some people take advantage of that and drill into it, and you can actually hit fresh water because it's less dense than salt water, so it occurs as pretty much a cell underneath the sand and on top of any salt water that might be under it. Typically have a back island dune field, tidal flats, and then we have in the respect of the Laguna Madre, we have some large areas that are algal flats, and then the lagoon and the seagrass beds. And these are just some depictions of, this is the back eye, or this is the vegetated flat behind the dunes, and this is I guess the fore island, and this, of course, these are coppice dunes here, and I can't tell what this one is. Oh, this is the algal flats behind that are in the Laguna Madre, and those are exposed by different wind directions. Usually our prevailing winds on the island are from the southeast, so they blow the water in the Laguna Madre is influenced more by wind than it is by tides, unless we have storm tides. The wind moves, if we get a north wind, the water in the Laguna Madre tends to pile up on the southern side of the Laguna. Does anybody know what drives all the currents in the ocean primarily?
This is Shark again. I would say density.
Robin got it. Wind. Wind drives most of the currents that we have in the oceans of the world. And when I learned that, I went, wow, that's amazing that wind can do that. It makes me wonder what is going on when we're actually harvesting the wind on shore now. And is it going to affect anything further, I guess, further on when we're harvesting more wind? Are we going to eventually slow down the ocean currents? I have no idea, but it's something to think about. This depicts the beach and dunes on South Padre Island. And if you notice the dunes, you don't see any real good coppice dunes there, because this was after a storm where they're showing the foredunes. They've been pretty much cut off by some very high tides. And then you've got the back shore, which has a lot of debris, or wrack, we call it. And does anybody know what is washed up on the shore there, on the foreshore, and mostly the foreshore?
This will be seaweed and algae.
Sargassum weed, and I believe it's a golden algae, and it forms the major, it comprises most of the Sargasso Sea. And Dr. Klein talked about eels that go out, the Anguillidae, that actually swim out and spawn in the Sargasso Sea. And that is where sargassum weed pretty much originates. It does attach in some places, but the majority of it, the source is the Sargasso Sea, which is in the center of the Atlantic Ocean. Boca Chica, which has recently been the focus of a lot of, activity from SpaceX, has some unique dunes. They have some dunes that are fairly large, and you can see some examples of the coppice dunes there in this picture. Right now, though, this was taken a few years ago. Right now, most of these coppice dunes have been washed away because of the hurricanes that we had in the Gulf of Mexico this last year, 2020, because they caused very high tides and high surf and were washed away. What happens to that sand when the dunes are washed away? Is it gone forever? Does anybody know?
No, it's not gone forever.
Where is it?
It washes back eventually.
Yes, that's right. That's right. It does wash back. And that's something, we think that we've got some real problems because the forefront of our dunes are gone, but it does come back. And there's some other things that I'm going to cover later on. Some places we lose dunes and we lose beach and it doesn't get replaced. Can anybody give me an example of what area that might not, or that isn't being replaced?
The south part of South Padre Island, where
That's
the
right.
where the county beaches are.
That's right, Kate. We have to have the Corps of Engineers dredge up sand from either the Brownsville Ship Channel, sometimes from offshore, and pump it back onto our beaches. And that's because of some levees or some jetties that we put in. Why do you think we have, and they were put into Brazos Santiago. Why would the jetties cause that sand to go away on the south side of the island?
This is Shredder again. This is due to the currents that carry the sand. They replenish the beaches, but since the jetties block that, it pretty much accumulates on the sides of the jetties instead of on the beach itself.
That's right. They interrupt the sand transport. This is just another look at the back dunes on South Padre Island, the grasses, and you can see the back dunes in the background there. So Shadrach was talking about what we call longshore drift, and that is the movement of sand along the coast. And if our wind is prevailing from the southeast, it brings sand. Which direction do you think it moves sand if our prevailing winds are from the southeast?
Just driving on the northwest.
That's right. That's right. And it is a general movement which goes whichever direction the wind is. And since it is coming from the southeast, the sand moves to the northwest or to the north along the coast, at least on South Padre Island. This is a drawing of the current direction coming from the north, and where you have the waves that are approaching pretty much from the, I guess you'd say, west-northwest, and they're driving that current, that longshore current to the south. And this is an important thing to understand, especially if you're a swimmer, and I'll get to that in a moment. We do have troughs or bays and horns or apices that form during wave action. And if you look at a beach, if you walk along the beach and look at it, you will see these curvatures in the beach, and that's due to wave action and current movement. So when your longshore currents are going in one direction, they start piling up water over the sandbars, the breakers do. And when it piles up enough, it starts moving it out offshore. And this is how we have formation of the apices, which are right here in this area where you can see it's building up and it's wearing away right here. So it moves out in a rip current. And remember I said that if you know which way the longshore currents are going, then you can save yourself a lot easier. What they do is they tell you if you're swimming and you're caught in a rip current, that you just let yourself go and go out in it, and soon it slows down and you can swim out. Well, if the wind is coming, say, in this direction I'll say this is the southeast, going from here, can you all see my pointer? To here, it's pushing most of the water toward the left side of the screen. Because of that, you don't want to try to get out of the rip current to your right if you're facing offshore. You want to swim in this direction and go with the prevailing longshore current that is moving. It doesn't move as much against the longshore current in this direction. I hope I'm making sense on this, but you want to go out in the area, in the direction that is usually in the direction of the wind and the longshore currents. I hope that makes sense, because during storms, though, all bets are off. If you get caught in a rip current, it's very tough to get back in during a heavy storm. You shouldn't even be swimming out there, really. So this is we have a unique, I guess, a unique situation on the Gulf Coast of Texas. We have prevailing winds from the southeast, and they hit the coast in a northwest, going in a northwest direction. But as the angle of the coast changes, it pushes the currents that are north of say the center of Padre Island. They push the currents to the south. So you have a current being generated from the southeast wind on the lower coast, and further up the coast, you have a current generated from the north. So what do you think happens when those two currents converge? They're both transporting sand, right? So I'll tell you. They're transporting sand, and it piles up. It piles up in that area. It is lots of turbulence, yes, Carolyn, but it forms a huge sand deposit, which in turn gets blown onto the mainland. And does anybody know about the sand sheet, or the Aeolian sand sheet, or the Aeolian sheet? Robin knows about it. It's the South Texas Border Chapter has had a lot of, I guess, presentations on the sand sheet. I don't know if this chapter has or not, but the reason the sand sheet is there is because that over thousands of years, that sand has accumulated where those two currents converge and blow on to the southeast or to the South Texas area right in here. Looking at sand dune growth, we have first of all the coppice dunes. If something starts obstructing the movement of sand, we have vegetation form, and then over time the vegetation moves or accumulates more and more and more depending on the wind velocity and the sand budget that it has. And then they stabilize as the beach moves in one direction or another. Beaches are not always in the same place. As you know, that South Padre Island was not there thousands of years ago. So the islands tend to move back and forth with sea level rise and sea level fall. Sand dunes are protective of our islands, of our barrier islands. You can see the pre-storm profile and the post-storm profile. And we mentioned earlier that sand tends to just move back offshore, and then it moves back in during calmer times after a storm. Usually we lose dune structure during the winter time, and during the summer, there's less wave action and the dunes tend to build back up because the sand is moved back on shore and then blown onto the dunes from the beach. So we talked about what happens at Brazos Santiago Pass. Shadrach said that we have a buildup here. You can see the sand tends to accumulate here. By the way, we just had a shrimp boat go aground in this area, and yesterday they just got it off the beach.
I actually saw that. I thought I was seeing things, but it must have took a few weeks.
Yeah, it was there for quite some time, and the guy that pulled it off was the son of Billy Kennan, and Curtis Kennan pulled it off with Billy's old boat, and they went into very shallow, rough water and just pulled it right out. That was an interesting thing to see. So here's South Padre Island, the southern end of it, and you can see that it's not getting the sand budget that it needs from the longshore current moving in this direction. So what's happening is it erodes away, so we have to replace it. And the Army Corps of Engineers usually does that. It costs South Padre some money for, to do that, to bring more sand in. This is an example, this picture, and I don't remember where this one was taken, but it shows typically, okay, what direction do you think the longshore current is moving along these beaches? Is it the same as it looks like on South Padre from Brazos Island to South Padre, or opposite?
No, it's going north.
It is, yeah, it's going north. Right. So what's out there? What's out there along the swash zone and inner beaches? This is my favorite shell. It's a lettered olive, and it is a predator that is extremely efficient in what it does. It has most of these shells. Somebody said, I saw something about erosion happening. I didn't see the rest of it. The comment. What did I miss on that one?
It says, is the erosion happening at a faster pace than seen in the past?
Yes, it is. It is happening that way, a lot faster on that end. On the north end, on South Padre, the south end of South Padre Island, the erosion is much faster than it was in the past. This lettered olive, I said it's a very efficient predator, and it moves through the sand. Does anybody know how these animals can move through the sand? I mean, I think it'd be really tough. Any idea?
The shark again. The sand is moist enough, or viscous enough that the animals can move through it.
That's right. There's a term for it, and I'll get to it in a minute. And it's really interesting that a lot of the animals that live in this area tend to utilize the viscous or the water mixed in with the sand, and what happens, and I'll get to that in a little bit. The sand dollars, they are related to starfish, and we usually find those on the second sandbar. And we don't see as many of them on South Padre Island as we used to, because there's so many people that walk our beaches that they usually get found before I even get to them. I've looked and I maybe found 2 or 3 over the years. Anybody else been able to find more? No comments? This is one of our more common shells that we find on the beach. And this, the moon snail, or it's also called a shark eye. And this is the egg case right here. It's mixed with sand. And these shells, the animal inside them can actually cover most of the shell. And you can see this one can also take advantage of moving through the sand. This picture I took, and I was amazed after I took the picture. We found this, I was south of here in Mexico, and this is one of our more common conchs that we find along the Gulf Coast. It's called the Florida fighting conch, but usually after a storm, you can find these on the beaches of Padre Island. But what was unique about it that I saw, I look at the eyes on it. There's one there, and there's another one. It looked like it was, this eye was looking at me, and this one was looking at something else. I don't know what. It was confused probably because I'd taken it from its home. but they're beautiful shells if you find them right after a storm. And if the animal is still in there, or if the animal maybe just passed away or died. The speckled crab is another dweller of the surf zone. You don't find too many blue crabs out there, which are more in the bay. And it's different on the East Coast, and they hang out more in the bay over here in the Gulf of Mexico. But these also burrow down in the sand. And I was wading one day and I stepped on there must have been 10 of them in one spot, and it was during their mating season. And it was very painful, to say the least. Somebody mentioned Washington Beach as a good place for sand dollars. So this is what makes sand an easy way for the animals to pass through it. It's called thixotrophy. And thixotrophy is the ability of a solid to turn into a liquid. And if you've ever been on the beach and you pat the sand and pat it, it gets more liquidy. That's what these animals use to pass through it, to pass through the sand, that ability. And it's one word I haven't forgotten it since I first learned it, and I think I learned it from Dr. David Hicks, who is with UTRGV now. But that's why what all these animals use they avoid predation, and they can also find prey in the swash zone. Swash zone is probably the most inhabited area on a beach. And we'll get to what makes up those inhabitants, or more of them. This is one of the prettiest occupants of the swash zone. These are very small. They're about a quarter, maybe they'll get as big as a half an inch. And they're called coquina or donax. And I think they have some other name for them. And call them surf clams. I haven't ever called them surf clams. I've only called them donax or coquina. Coquina soup, Linda mentioned that. Coquina soup is really good. What they do is they'll collect coquina shells out of the swash zone. You can see them come to the surface sometimes by the hundreds and just pick them up. But you have to let them sit in water for a while to let them pump themselves out, because they have a lot of sand inside them. And then you boil them and then throw the little clams away and use the soup, or what do you call it, broth, you can make, you can add whatever you want to it, but they're quite tasty. These are beautiful little shells, and we only have 2 species that occur on the Texas coast, Donax variabilis and Donax texasiana. These are the siphons here, but they also have a foot. You can see the siphons. Siphon brings in water and lets water out. And here we go. Here's how they dig into the sand, and they move with the swash zone. They move inshore during a flood tide, and offshore during an ebb tide. And these are the 2 siphons right here. And this is the foot. And this is how they move in to the sand. They stick the foot in, and then they bring water into their foot, and it expands. And then they pull with their muscles and expelling some of the water. They pull in. And Robin asked if you can harvest them. Yes, you can harvest them. There's thousands and thousands of them out there. And that's how they disappear and hide from predators on the beach. They also surf. They call them surf clams because they do surf. The shape of the shell, they're thicker on this end and thinner on this. So they will face the waves in a certain direction. If they want to move up the beach, they'll get on the surface of the sand, and a wave will come in and it hits them and moves them hydrodynamically up the beach or down the beach if they want to go. It's very interesting to watch them surf in the swash zone. This is another interesting predator that we have, and it primarily eats some of the smaller animals, and it's called an auger. And they're not very big, but they utilize a sail to move up and down in the swash zone. And the sail is their foot, and they spread it open when a wave comes in, and they actually will move up the beach. And then if they want to move back out when the tide's going out, they'll use their sail to go in another direction. Have you ever seen the little volcanoes that are in the water when you're walking around in the shallows along the beach when it's not too rough, or even when it's rough, but when you can see it, they look like they're even I guess letting or jettisoning sand. What is in there are ghost shrimp, and these are some of the best bait that you can find around the beach, because everything loves to eat these. They're very soft shelled, and you can even see the egg case. This is a female right here, and they're always cleaning out their burrows, and you think, oh, there's just a little tube down there, but no, this is what some of their burrows look like. So this is the surface of the sand right here, and these are the burrows. And there's also other animals that coexist with them in there. Pea crabs. There's some pea crabs that live with them. What kind of relationship is that? Parasitic, commensal? Does anybody know? You got it, Shadrach. It's mutual. And these burrows can be at least sometimes 6 feet deep. And the way they studied them, they poured plastic into the burrows to figure out what shapes they were, because you can't really dig in the sand because of thixotrophy. But if you pour something heavier or even metal, you can pour it. They've actually poured metal in them. One of the other baits that people talk about for fishing for surf fishing anyway, is mole crabs. They're also called sand fleas. If anybody reads about fishing and they talk about sand fleas being bait, well, these are what they are. They're a crustacean, and they're about, I would say maybe they get up to almost 1.5 inches long, and they're almost shaped like a small lobster. Their tail is tucked in. This is the tail section. And the way they feed is they burrow into the sand and they stick out their antennae, and they depend on the surf when it's on the waves, when the tide is or when the wave goes out in the swash zone, they capture any animals or algae or what have you that may be there, and they take it into their mouth. And this is one in action right here. You can see the direction of the movement of the water is probably going this way, because the antennae are facing in this direction. Amphipods. Somebody asked, was that a question? I didn't see it. I'm sorry. Can someone tell me what the question was?
Are the mole crabs common?
Yes, they are very common. I'd love to do a field trip down on the beach, because it's the stuff that is in that area in the swash zone is amazing. Speaking of amazing, this animal is the most populous animal along the swash zone. And probably worldwide, there are more of these than any other crustacean. And it's an amphipod. And these are tiny little things. They're less than 1/8 of an inch long. and they live in the surf zone. And I mentioned the auger, and it feeds on these. Pretty much anything that, or any of the animals that are a little bit larger than the amphipods will feed on them, but they're out there in the millions and millions and millions. So let's talk about the strand line. That's where the waves have deposited what we call rack washes up on the beach. And of course we have sargassum here. And then these are tubes to ambush predators. And they're worms that live in the sand. But these have been exposed by the waves and washed up on the shore. Does anybody know what this animal is? It's a fish. It's a sargassum fish, and it lives in, no, it's a sargassum fish, Eric. It lives in the sargassum weed. And sargassum has its own ecosystem of animals that mimic the sargassum weed, so predators can't find them. You can barely see that that's a fish. It's a member of the anglerfish family, and I don't know if Dr. Klein covered that or not, but they have a small adaptation right in front of their eyes that they wiggle, and it looks like a little worm or something, and a small fish will come up and try to eat it, and they won't see that this is a larger fish. And the anglerfish or the sargassum fish will swallow the little fish that's trying to eat the appendage that is around its eyes. Portuguese man-o-war is a colony of different animals, and the cells in those have different I guess methods that they utilize the prey that they catch. They actually have tiny harpoons in some of the cells, and these are the stinging cells. And there's also triggers on those cells. The harpoon is embedded into the cell, and the trigger sticks out a little bit. And if a fish rubs against that little trigger, the harpoon will stick out, will shoot out, and it looks like a harpoon, and it's attached to the cell. But when a fish comes by, thousands of these cells fire harpoons into the fish, and they can pull the fish back in. And then they bring the fish back up toward the stomach, and a different set of related cells work on it to digest it. And then another set of cells form what we call the sail in here, and it has a bladder filled with gas. Sea turtles love to eat these jellyfish. I call them jellyfish, but they're hydrozoans. Just recently, we've had some of these wash up on our beaches on South Padre. I don't know if anybody has seen those. These are related to the jellyfish. They're called Velella, and they're by-the-wind sailors. And so, Molly, you saw those, I guess. Well, that's good. Usually during February, we have animals that float around in the Gulf. They tend to wash up during this time. We've had thousands of Portuguese man-o-wars wash ashore during this time. Another one is porpita, or the blue button, they're called, and they're really pretty. They're about the size of a quarter, and they'll wash up too. The purple sea snail is a predator on the Portuguese man-o-war, and here's one attacking a Portuguese man-o-war. And I found some of these. Their shells are paper thin, and you're really lucky if you can find one that is intact. but they're quite the predator on these jellyfish. One of my favorite jellyfish is the cannonball. They call them cabbage heads, and there's other names too. But this is an important commercial species, and people love to eat it. Does anyone know where they might eat these jellyfish? Where people might like to eat them, and how they eat them? No one? Well, Japan is a primary consumer for these. We tried to establish a fishery for these off Texas because sometimes they'll just be thousands and thousands of them in the water. And we were trying to give fishermen another opportunity to be able to harvest these, but the Parks and Wildlife said no. But in other areas in the U.S., they are harvested and they are sent to Japan. And these get as big as a cabbage, as a large cabbage. And what they do is they slice them up and they treat them somehow. And I've eaten them in Japan, and they almost have the consistency of a soft rubber band, but they're delicious. And the ones I had, they were kind of yellow mustardy color, but they really are good to eat. And I never thought I'd like to eat jellyfish, but now I hear that there is a drug that they extract from jellyfish, and maybe they use those for that too. That helps you with your memory. So this is a barnacle, one of the barnacle species that we have along our coast. It washes up on the beach. It likes to attach to something hard, and it's a gooseneck barnacle. And someone once defined a barnacle as an animal that lays on its back all its lifetime and kicks food into its mouth. And these are really interesting animals because they also have a parasitic species of these, and I'll show you that. It preys on the blue crab that is an important fishery here in Laguna Madre. The blue crab is very popular in the Chesapeake area. Its name is Calanectes sapidus, beautiful swimmer, and sapidus, I believe it means sweet. So, and it's the favorite crab meat that my favorite that I've ever tasted. The way you can tell the difference between the two, the male and the female. This is a male. The male usually has blue tips on the inside of its claws. And I used to tell kids when I talked to them in school that the female had fingernail polish. But then one day, some of the kids held up their hands, and the guys had fingernail polish. So I can't say that anymore. So this is the parasite that is part of the family of gooseneck barnacles, and it attaches to the gills in the blue crab, and it interferes with respiration. And I guess it was last year, our crabbers reported that their crabs were dying when they pulled, or they were already dead in the traps when they pulled them up, or when they put them in cold water to slow down their metabolism so they could ship them off to, say, the Chesapeake, because that's a major market in the US. They would die when they put them in the cold water. And so we looked at the crabs, and they said they had found some things that looked like rice grains. They were tiny, and that, lo and behold, that's what it was. We sent it off to Texas A&M University, and they identified this as being one of these things, a gooseneck barnacle. And it only likes really high salinity water. And we had high salinity early in the year during 2020. But after these storms started appearing in the Gulf, we started having more rain, and the incidence of that barnacle went down. And the crab population is still depressed, but hopefully we won't have some high salinity, or we are getting into a drought, so that it might come back. We really don't know. We have 3 commercial species of shrimp in the Gulf of Mexico, and the size of these doesn't mean anything. They get up to, all of them get up to about 8 inches maximum length. And this one right here is a white shrimp species, or the white shrimp species, and it is caught usually near shore, and it doesn't comprise the majority of the catch of our commercial catch. Our commercial catch depends primarily out of Brownsville, Port Isabel on this species, which is called the brown shrimp. And they're all members of the Penaeid family, the family Penaidae. And then this is called the pink shrimp, and some of the fishermen call them hoppers because they're more active when you drop them out of the net onto the deck. They jump around a lot, and they're really good bait because they're an extremely hardy species. This is a minor catch. We have more of these that occur off of Port Mansfield and some of the sandy areas. And then the primary fishery for these is off of Florida Keys. But brown shrimp comprise most of the catch off Texas and Louisiana for our shrimpers. And then off Louisiana and the upper coast, the white shrimp are the most, I guess, most common that they catch. In Brownsville, we catch about $70 million worth of shrimp annually. Very important fishery. We have around 185 shrimp vessels that pursue these. So I talked about sargassum earlier. We do have 2 species of it. Some years it washes up in tremendous amounts on our beaches. And why is that so? We think it's because of all the nutrients that come out of the Mississippi River. Does anybody know what other impacts the nutrients have in the Gulf of Mexico besides fertilizing this algae?
Harmful algal blooms. It struck again.
Well, not so much harmful algal blooms, but we have a dead zone that is thousands of square miles in the Gulf of Mexico that forms annually. And they think it is because of all the residual fertilizer and nutrients that come down the Gulf, and the nutrients are utilized by algae, which uses up all the oxygen in the area, and then everything dies. And some of the old time shrimpers used to report that they weren't catching anything off of Louisiana, and that was the reason why, because of the dead zone. And this is another sargassum fish, and this is some sargassum that you usually see it floating in large concentrations like this out in the Gulf. And when you see this, if you fish, you want to fish around these because fish like to, or predatory fish like to hang around it. And so that's one of the secrets that you learn fishing the Gulf of Mexico. If you see something like that floating out there, you want to fish around it. And I think all these species were covered by Dr. Kline. The bonnethead shark, I don't think he mentioned that one. It's a member of the hammerhead family, and it is not just a carnivore. It can be herbivorous, and it eats seagrass at times. And we just learned that through some of the research. And that's very unusual because most sharks, or all sharks, I thought were carnivorous or all the time, and they're not. This is a unique species. This Florida pompano occurs off our Texas beaches. And Kate, I think you caught one of these. Did you catch one? Or maybe not. I don't know.
No, that wasn't me. That was Richie.
Oh, okay. This guy that we fished with, he caught one that was about 3 pounds, and they come up to spawn off of Texas annually, and the run occurs usually during midwinter. And I think it's just starting to fall off right now. And that's all I'll say about the fish. Oh, we've got some whiting. They're a member of the family Cyprinidae too, and I think Dr. Klein covered that. And they talk to each other via their I guess, the noises that they make in order, if they want to attract a mate, they will make these noises. I've got some more. Oh, here's a spine to, he talked about the stingrays. This is one. What do you think happened to this one right here? A shark.
Yeah, shark came after it.
So this is what the spine looks like on the tail of a stingray. And I've looked at some shark mouths and I've seen stingray spines stuck in all over the inside of the mouth. You think that'd be extremely painful, but I don't know. They think that fish feel pain differently than we do. I don't think I could have stood that, but this is a serrated spine and it's covered by a bacterial sheath. So you not only get wounded, but you can get an infection, and it has a toxin in it too if you get stuck by one of these. The toxin is based on protein. So one of the, does anyone know how to denature a protein?
Well, this will be heat.
Heat, that's right. So if you ever get hit by one of these and it's not bad enough to go to the hospital, if you just have a wound and it's extremely painful, I've been hit by them, you just immerse the area that you're hit. Meat tenderizer works too, Robin, but you can immerse your foot in, say, a 5-gallon bucket of hot water, and it'll dissipate the pain very quickly because it denatures the protein. So one of the things I wondered about, if a lot of people are fishing out of outboards that wade the bay, and how would they get hot water way out there? Well, outboard motors use water to cool themselves, or the way they are cooled is by water. So they actually have a spout that shoots hot water out. So if you have a bucket, you could actually use that water to treat the wound. And I don't know if Dr. Klein covered this species. This is part of the mackerel family, and they get up. This is about an average size one, and these are absolutely delicious to eat, Spanish mackerel. And they usually occur, they have big schools of them come around here during offshore, near shore here during the fall. I'm sorry I've got so many fish in here. I didn't think he was going to cover all of them, but he covered quite a few. Sheep's head. Right now is the beginning of sheep's head fishing, and off the jetties you can go out and use small pieces of shrimp, and they say that you have to jerk on the pole before the fish bites it in order to catch it. That's what I was told many years ago. They have teeth that almost look like human teeth. Can you see that down there? And they use those for grazing on barnacles along the jetties. And also they can eat crabs very easily. These are two other cyanids that occur, the croaker and the spot. And the spot is called a spot because it has a spot right here. And these get up to about, the croakers can get up to 5 pounds. And the largest spot I've ever seen is about 1 pound. But they're all good eating. They're all cyanids, except for the sheep's head. I don't think it's not a cyanid. So here's the food web on the beach. Small particles and decomposed plants are taken in by filter feeders. And one of the filter feeders I mentioned was the coquina, and predators in turn eat those. Food from algae, jellyfish, blue bottles, or small particles that are washed up. Scavengers eat those. And then the predators, the seagulls, fishes, and crabs. One of the saddest things I ever saw was in Mexico. They were doing a baby turtle release, and it was a political release. The governor of Tamaulipas was there to show off their program that they have to raise to take the eggs out of the nest and transplant them to protected areas to grow up. Well, then he was going to release them, and seagulls were diving on the and eating the little sea turtles as they release them. Very sad to see. Somebody just made a comment and I missed that one too. They're not staying up long enough. Did anyone see it?
It says, no, just use a small circle hook and they hook themselves basically, but they do love to tangle you up around the columns wherever you are fishing.
Oh, these are the sheep's head. Thank you. I often wondered if circle hooks were effective for those because they're such delicate biters and great fighters. And of course, we have sea turtles that lay their eggs on our beaches. The Kemp's Ridley, we're getting ready to start the season right now. We also have green sea turtles that eat sea lettuce on the jetties at Brazos Santiago and Port Mansfield. And they're usually juveniles, but these are adults that come in and lay their eggs. The Kemp's Ridleys are the adults, and only the females come ashore and lay the eggs. So I'm going to talk a little bit about one of the algae. Shadrach, you mentioned harmful algal blooms, and this is probably the most harmful one that we have off our coast here in Texas. And it occurs infrequently, pretty much. You can't really predict when it's going to happen. It's always out there. It's called an unarmored dinoflagellate. It's large. It's easy to find it under a microscope. And we have trained red-tide rangers that can identify it and count the cells. And they can also measure the amount of aerosol that is in the air. Because these aren't unarmored, they tend to break in wave action, and they release a strong neurotoxin called brevetoxin, and it can irritate your nose, it can affect your breathing. People that are asthmatic should not be around beaches when we have these blooms that occur. Joni, you're a red tide ranger. Can you tell them a little bit about your experience?
Sure. I know that it took a lot of practice to be able to identify the cells when we were looking at it through the microscope. But now with the software that they have, it's a little bit easier. And counting can drive you a little stir crazy, because that's all you're doing is sitting over a microscope counting the number of cells. And I guess too, Tony, you could add that there's a certain count that you're looking for to know whether there's a possibility of a red tide, correct?
Right. If it gets, I guess, between 2 to 5 cells per milliliter, that's when you need to watch it, and you could possibly have that happen. So
right. So the way it works is people are sent out to collect water samples in different areas around the island, and then those of us in the lab would take some of those samples and put them on slides, and again count the number of cells, and you had to do it for a certain measured area, count the number of cells.
Yeah, and it's a lot. I enjoy it. I like looking at these cells. They're really interesting. The ones here in this slide, you can actually see the nucleus. It's been stained, and we use a stain to kill the cells and be able to count them more easier. Yeah, but it's a fairly easy, I guess, algal bloom to identify and to count the cells, because it's pretty much the only thing that's out there when it is there. Sometimes during the fall, we may have other blooms, but they're real easy to pick out. We have a new device that Joni mentioned. It's called a HAB scope, and we actually put a drop under a microscope, and we have a device that can track the cells, and it uses facial recognition to count the number of cells per milliliter. And they're getting better and better at it too. So it's going to, hopefully it'll take a lot of work away from us. But the National Oceanic and Atmospheric Administration depends on the red tide rangers during a red tide bloom to actually track it. And they use the cell counts, and they can track the bloom, the direction that it's moving, and they can actually predict what's going to happen now, or whether it's going to go away, or whether it's going to move north or south. And it's really interesting. And the Red Tide Rangers actually won a Gulf Guardian Award in 2006 from some of the work they did the previous year.
95 mask up with the aeratized red tide.
Yes, yeah. In fact, even the cloth mask help because it aerosolizes in larger droplets that, especially during high winds. But I guess the N95 would be the best because it removes 95% of the aerosol from the air. And I just read something about cloth masks versus the N95s, and there's a 94 from Korea that are much better. It removes 94%, but the cloth mask, they say, only remove like 45 to 50% of the aerosol. So you're really risking it wearing cloth mask. And I just read that today.
Excuse me, sir. Since K. brevis releases toxins since it doesn't have a shell, theoretically, if the dinoflagellate were to have a shell, would it become harmless?
Well, it probably wouldn't break up as easily in the surf. There's dinoflagellates that are armored, and you don't see many broken ones when you have heavy surf, and you can actually see pieces of Karenia brevis in some of the slides. Sometimes they'll even lump together. So yeah, the armoring would prevent it, but I haven't heard of any armored ones that have a toxin in them like this one does. This one may have that toxin in it to actually feed itself because it kills fish. The toxin kills fish. So it may, and it's been theorized that the fish die and it feeds not on the fish, but on the nutrients from decomposition of the fish. It's part plant, part animal. So it depends on sunlight to make its own food, but it takes in nutrients because plants utilize nutrients and the sun also. But this dinoflagellate can actually move through the water because it has flagella on it. So it acts like an animal, and it is part plant. It also kills mammals. Ground squirrels, one year we found 10 dead ground squirrels, coyotes, even dogs can breathe some of the foam that is produced in the waves from all the cells breaking up. And here's somebody wearing a mask, and those are so helpful. Somebody had a question?
Yes, Tony, it's Diana again. Do you have to test the toxicity levels in the water, or is this mainly an airborne toxin?
We just do the airborne toxin. If you did it in the water, it'd be a really tough, or probably a hard way to determine that, because you'd have to maybe it would take a lot of probably research to even, or not research, but maybe some machine that could determine whether it was brevetoxin that's in the water, and then to determine the amount. The way we determine it is if we're, say, coughing a little bit, or our eyes are watering more, or we're coughing a lot, or we have a sore throat, or can't breathe very well. It's an individual judgment, but it's like, you think it's like a low, medium, or high concentration. And isn't that how we do it, Joni? We measure it that way?
Yes, that's right.
Then
Yeah.
do you close the beaches determining by that? Or does someone close the beaches to keep people away from it? Or what?
No, the county usually will put out a notice for animals, for pets, because coyotes, they think, may die not from breathing it, but they eat some of the fish that accumulate it, and they gorge themselves, because they don't get food all the time. They'll eat anything. So it's people pretty much will go out and they'll feel it and they'll leave. It's self-regulating.
Well, I'm from Kansas, so I helped develop the harmful algal bloom program there in Kansas.
Okay.
And so I'm familiar with blue-green algae. Unfortunately, people in Kansas don't regulate themselves and they go swimming in it sometimes. But that's why I was asking, because we had to test the toxicity levels, and then we determined from those toxicity levels whether we had to close a lake or not.
Yeah, those blue greens are, I guess they're a different sort of animal because they don't produce an aerosol. And
they can, but there's research out as to how far that atomizes out into the air. We're not sure yet.
But this aerosol, it's there, and you want to leave if it's out there. So like I say, it's self-regulating.
Yeah, I have to say that one time, it was during the sand castle, when they were building the sand castle and the competition, and I remember going out there and I had to leave. I could not tolerate the toxin, yet there were people who were wearing masks. So it's really up to the individual who can tolerate it and who can't.
That's rough.
Yeah, I know I couldn't.
So it does cause fish kills. The fish absorb the toxin through their gills, and they pretty much get paralyzed and they can't breathe and they die because it has quite an effect, more of an effect on fish. And some of the fish, they like menhaden that go through the water and feed on algae are usually one of the first ones to go, and they're like the canary in the mine. If we see a lot of menhaden on the beach or a fish floating in the water, then we know we might be having a red tide and we'll start sampling. Those fish kills are probably one of the first things that we notice, and it killed these rays. These butterfly rays were killed by it, and I guess red snapper. This is a gaff top. And here's one of the Mexican green bleach bottles that float up on our shore that I mentioned earlier. So what else is out there? There's earless lizards. There's the rattlesnake, and there's also, I think, there's just going through some of the info. Eaten by coyotes and ground squirrels. And so here's a 6-line one, and this is the cutest one out there in the dunes, the kangaroo rat. And a friend that he was going to study those for his master's, and he never got around to it. And I was hoping he would, because they're just really interesting, and they're preyed on by many different predators out there in the dunes. Nocturnal, eats seeds, has cute little cheeks when it fills them up. Doesn't drink much water. It gets most of its water from the food. 3 to 4 young. Large hind legs. It jumps like a kangaroo. And total length is 8 inches. That's from the tip of the tail to the nose. Weighs 1 to 2 ounces. Wow. The ground squirrels, the spotted ground squirrels, I told you that they were, we don't know how they died from the red tide, but they did find brevetoxin in their, I guess, internally. So they did detect it. And a Dr. Zimba at Texas A&M Corpus Christi did some of the studies on some of the dogs that died from it. He detected the brevetoxin. It's quite a sampling technique to find that. They don't venture far from their burrow. Coach whips. These are a really cool snake. They get long too. They're I've seen them maybe 7 feet long. And the coyotes, they're always out there. They don't hunt in packs. I didn't know that, you know, until we usually just see single coyotes. But in the morning, if you've ever spent any time in the ranch land, you can hear them howling. And you wonder if they must all be together, at least when they're in their dens. And the birds, I learned that if you see a fairly large bird on the beach, that it's probably, I think it's a willet. I'm not much of a birder. The red knots, I did do a study once with UT Brownsville, where we were looking for red knots. Red knots are migratory, and they tend to pass through here, and they like to spend, I guess, their summers up around. Did somebody ask? Someone brought up, I made a comment and I missed it. I don't know why I'm missing all these. Anyone noticed it is now after 9? Oh, it is. I'm out of here then. The birds migrate and I'm going to migrate too, because there's a lot more left. So.
Can I ask one more question about the man of wars, the Portuguese
Yes.
man of war? Because they're so common. And I was wondering, when the small ones are on the beach, how dangerous are they? Because there are thousands of them at times and they wash up after a storm. And second question, in the summertime, when you don't really see them on the beach, is that what you get stung by? If you get a sting when you're in the water, deeper in the water, is that from a Portuguese man of war?
Not if you can't see it. You usually see the sail on them. If you don't see one around you, they can have tentacles as long as, I think I read 60 feet one time.
So
they're usually a
so
smaller.
are they just more a winter thing on South Padre?
More so when they occur in large numbers and drift up, yes. During like February is the time they usually come ashore, but they're always out there.
Okay.
And it's not those that are probably stinging you during the summertime. We have some other pretty nasty smaller jellyfish that you really can't see very easily, and they have some very powerful stings. The man of war has a fairly good sting. I've been stung many times by most of the jellyfish. I think the moon jelly is probably one of our more common ones that we have around here. And it's just as painful to me as the Portuguese man of war. Some people are more sensitive to say the man of war than others.
Okay. Thank you.
You're welcome.
Okay, Tony, thank you very much for the presentation this evening. Sorry if we cut into your time with our little break, but hopefully.
It wasn't your break.
But hopefully maybe we can have a field trip at the beach once we get past all this COVID stuff. That would be nice.
I hope so.
Yeah, so do I. Thank you again. Did anybody else have any other last questions for Tony?
This is Kate. I realized earlier I put my email message in a private message to someone. I didn't share it with the whole group, so I put it in to everyone. It's a few comments back, but anybody from the RGV chapter, my email's there now.
There's Joni.
Oh, no.
Great. So how do you sign up for the Red Tide Rangers?
Well, we usually have trainings. We put them on Facebook. We have a Facebook page that
Okay.
you can, if you like us or I guess follow us, and you'll get a notice when we have trainings. We had 3 trainings last year because of COVID, and they were primarily for Red Tide Rangers that had done it. I think Kate went, didn't you? Yeah, Kate was at one of the trainings.
Yeah, the last one you had, it was like November or something. There's only 5 of us there.
Yeah. So just check the Facebook page, and hopefully we can do more.